Okay, hello everyone. Welcome to another episode of DrugBank in Conversation. Today we're joined by Bryce Lefrancois from DNA Genotek and Diversigen. My name is Chris, and I lead the scientific support team here at DrugBank, and I'll be your moderator today. Today we're going to be discussing lessons learned from longitudinal modulation of the human gut microbiome and the implications for clinical studies. So our presenter today is Bryce Lefrancois. So a bit about Bryce and Genotek and Diversigen. So Bryce is a senior R&D manager, and he leads a team working on novel technologies across both DNA Genotek, which is mainly focused on devices, and Diversigen, which is mainly focused on services. His team has spearheaded the development of novel tools and workflows, as well as innovative preservation chemistries to facilitate multi-omics analysis of gut microbiome samples and beyond. So, since joining the team, he has worked extensively on optimizing the stabilization and processing of human microbiome samples and has acquired significant expertise in study design, selecting of appropriate controls, and workflows to generate insightful and reliable data. Prior to joining DNA Genotek, he obtained his PhD in biochemistry from the University of Ottawa and has over 20 years of experience in research across a wide range of fields, including cancer and neuroscience. A bit about the companies. DNA Genotek and Diversigen are both subsidiaries of OraSure Technologies Incorporated, offering high-quality microbial sample collection products and comprehensive services for sample processing, sequencing, and bioinformatic analysis. So very briefly, today's webinar will consist of a roughly 40 to 45 minute presentation that Bryce will give. Then we'll have about 10 minutes for questions and answers afterwards. So if you have questions during the presentation, please feel free to pop them into the chat or using the question function in the app here. And we'll do our best to address as many of them as we can during the Q&A session. And we'll have about 5 minutes at the end for wrap up. As one final reminder, this webinar is being recorded and will be shared with all attendees and registrants via email after the event is over. So without any further ado, let's get started. I'll pass things over to Bryce for your presentation, and thank you so much. I'm really looking forward to it.
Thanks for the introduction, Chris, and thanks for everybody that's tuning in today. I'm going to be walking you through a lot of the data that's been generated through both DNA Genotek and Diversigen, and kind of proof of concept data about modulation of the gut microbiome and how to basically detect changes when you're trying to alter the gut microbiome. So I think the past decade or so has seen tremendous progress on the microbiome research front. And there's been an evolution of the technologies, and we've seen a lot more personalized microbiome characterizations tools. So a lot of people can now access kits and tools to know more about their own microbiome. And the technological improvements have been expanding sample collection, processing, preparation of the samples. There's also been a decreased cost for sequencing. And all of that has made the perfect storm to really generate more and more data and personalized data. And a big part of that technology advancement is also around analysis, databases, as well as annotation tool. And I think through all the research that has been done, so it's impossible to give credit to all the great work that has been done. There's really been a lot of potential link that has been established between health and disease state and the human microbiome. And one of the most studied sites is the gut microbiome by far. And I think right now we all know that it plays a central role in human health and diseases. And it's been tied to IBD, so irritable bowel disease, Crohn's disease, but also the gut-brain axis where the gut communicates with the brain and it leads to better outcome. I think what's interesting is that if you take a thorough look at the literature, most of the evidence to date that we have on the human microbiome has been based on metagenomic sequencing, so it's DNA based. And one of the questions that a few folks have started to ask, and that we're asking ourselves as well, is it always the most relevant analyte to look at? And that's where we come to, well, what are like basically what is the best analytes and what are the differences between DNA and RNA and metagenomics versus metatranscriptomics. So DNA on its own is like really a great way to like identify the species that are present in the sample. So that's kind of community membership. And it's relatively easy to stabilize, to extract. So I think it's been a go-to for a number of research studies. And what's interesting about the DNA is that with like the current databases we can now like predict like the sample functional potential. So look at the genome and all the genes that are like present in that metagenome and basically everything that could be expressed by those microbes. On the other end, I think RNA, there's been less work around RNA because it's like much more difficult to work with. I think everybody here probably knows that RNA is incredibly unstable and degrades very readily. But I think one of the main appeal of RNA for like microbiome research is that it's actually going to give you the full picture of the microbial cells that are active in your sample. And it can also, like by measuring gene expression and metabolic activity, like tell you what is being expressed and what are the bacteria doing in like a particular environment. And there's been a few studies, I think, that have looked at gut metatranscriptomics. It's still like early days for that field, and I'm highlighting like 3 here on the left side of my screen. And I think overall, they all come to the same conclusion. So generally speaking, if you compare the metatranscriptome to the metagenome, it's much more dynamic. And that's kind of expected because we know for a fact that gene expression like changes much more rapidly than genome can change. One interesting conclusion of most studies that have done like gut metatranscriptomics is that abundance as per like DNA-based sequencing does not always equate activity. So you can have cells that are highly abundant when you do like metagenomic sequencing, and yet they're like dormant and inactive. So they express very few, if any, mRNA. Then there's also the concept of like core versus the variable metatranscriptome. So kind of the housekeeping gene that every bacteria needs to express to like go about its day, versus like the variable metatranscriptome, which is really like the function that are expressed in response to a particular environment or a particular cue in that environment. And most interesting, I think, is that with all the data that we've generated, we've seen that if you look at the average microbial sample, you'll see like multiple species sharing the same metagenome, so the same functional potential. And yet, when you look at who's active, you'll notice that there's only a few species that are active, meaning that despite having several bacteria with the same potential, they're not all kind of occupying that space and being active at the same time. And I'm presenting some data here that was generated in the very early days of our work, where we took like samples from adult and infant samples and kind of looked through metatranscriptomics at the active taxa. So those are taxonomic bar plot. And as you can see, all the donors are like showing a lot of differences. So there's a large diversity in the number of active taxa. And if you compare the samples between themselves, so you're kind of looking at dissimilarity through like the breaker disk and looking at taxonomy only, you see like the value is close to 1, meaning that each of the sample is unique and very dissimilar because it's not the same species that are active or present. What's interesting, though, is that if you look at the express function and the KEGG orthology and do the same analysis, you see that you end up with a breaker disk dissimilarity value that's much lower. So we're talking about 0.2. And that means that the samples are much more similar. And that's kind of the concept I think that's important for like gut metatranscriptomics, is that there's a significant convergence of the functional output. So you may have different species, but they tend to do the same thing. And the reason behind that is that they occupy like the same ecological niche. So you need like a certain set of transcript to be happy and functioning in that niche and like use whatever is in the environment. So metatranscriptomic in general is very attractive because like it actually captures like expressed gene and can help us like distinguish active versus inactive bacteria. It also like captures that dynamic nature of gene expression, but also that kind of opens like a world of possibilities where you can look beyond bacteria and start looking at, for example, the host or like RNA viruses. And I think that's important, especially when looking at health versus disease state. But there's many challenges that come with working with RNA. So as I kind of mentioned, RNA, very unstable. And to work with it, you really need like optimal means of stabilizing the sample, but also optimal means of processing it, because you want like pristine RNA to like maintain as much of the information as possible. Also, like when you extract RNA from a microbiome sample, the vast majority of that total RNA is ribosomal RNA, so that's part of the ribosome to translate protein. And ribosomal RNA in itself like contains like very, very little information or very little useful information, and it actually is not a representation of the active species, so you would want to remove that as much as possible to get more insights into like what's expressed, so the mRNA. And also, as mentioned, like annotation is probably very important. Most people have been doing metagenomic sequencing, working with genome. So I think if you want to be looking at metatranscriptomics, you would want to like build a solution for the ground up for annotation and really dig into the express genes and function. So the way we approached this was like multi-pronged, and the first thing that we did was tackle sample collection. And that was mostly done through like DNA Genotek, because like they have a lot of expertise on like sample stabilization and processing. And if you want to recover high quality bacterial RNA that's going to be representative of the in vivo state of the sample, it's very difficult. So stool is particularly difficult because you're dealing with very high biomass, a highly complex community with like hundreds of species, but also I think something that few people recognize, very high level of nucleases. And just to show you how much RNA's activity you're like dealing with, we did that simple experiment where we measured RNA's activity in stool sample using like a probe-based assay, which is called RNase alert. And as you can see on the graph that's on the right side, there's very significant activity in stool sample, generally speaking. So you really need a solution that's going to like prevent that activity. Also, one of the tricky thing about bacterial mRNAs in general is that they have very short half-life, and bacteria by design are like meant to like quickly respond to change in their environment. So things can change like very rapidly, and you don't want the collection nor the stabilization process to like kind of skew your profile. So we came up with the OmniGene gut DNA and RNA about like 1 year ago. It's been commercially available for 1 year, and very quickly just kind of showing you the performance of our device for like stabilization of DNA and RNA in like the same tube. As you can see, we see good stabilization of both DNA and RNA for weeks at room temperature. If you like store the samples for up to 31 days, there's a bit of degradation, but generally speaking, we see much better performance than some of the competitor solutions that are on the market, where you do see that they do a good job at stabilizing and preserving the DNA, but you do see significant degradation of the RNA within days or like week of sample collection. Also, the OmniGene gut device is meant to be used for at-home collection. So the idea and the use case is that somebody can collect in the privacy of their home and then ship that sample back to the lab for like processing. So we also tested the robustness of our chemistry and device to like sustain the condition that are like encountered during transport. And we subjected like 4 samples from 4 different donors to like cycles of freeze thaw, so where we're like doing -20 to like +37. And as you can see, even when we put the samples through those harsh conditions, we do see very good stability of the RNA and the DNA. And that's not the case for like some of our competitors, where you basically start to see like degradation in the case of donor 2 and 3. The next thing that we needed to really tackle as part of gut metatranscriptomic was the nucleic acid extraction part. And extraction, I think, in the microbiome field is crucial for like accurate profile recovery and knowing what's in your sample. There's tons of data that's been generated over the past few years that has shown that mechanical lysis is critical to capture both gram-positive and gram-negative bacteria. But when we did some early testing and kind of benchmarked different extraction kits, what we actually noticed is that they were all performing very well for the DNA, but there were like drastic differences in performance for RNA. So we tested here like 3 kits, A, B, and C, and co-extracted the DNA and the RNA. And as you can see, the performance and the quality of the recovered RNA like varies quite a bit kit to kit. And back to my comment about that high level of nuclease activity in stool sample, you need to make sure that you basically pick a kit that's going to be able to mitigate the impact of the stool nucleases during the extraction process, and failure to do so could result in degradation that's happening as part of the processing, no matter how well the samples was preserved and collected to begin with. So very importantly, and I think the main point here is that stool metatranscriptomics does require the highest starting RNA quality possible to like really maintain as much of the information as possible. The last step in the process is the processing of the samples, so library prep and then sequencing and analysis. And here I'm kind of highlighting the metatranscriptomic pipeline that was put together at Diversigen, and that's a service that's readily available. And as you can see, the first thing that we had to make sure is that we were going to do effective removal of the ribosomal RNA content, because this is not something that has much value. And as you can see on the graph on the left hand of my screen, there's very little ribosomal RNA that's left. It's like effectively depleted during library prep. And as such, you really maximize the coverage of that coding content. So that would be essentially the blue bar. And most of the reads are like that kind of coding material and those functions that are expressed. We also did a lot of work around optimizing the sequencing depths. So sometimes metatranscriptomics required like pretty hefty depths to like get after like lower abundance function and gene and transcripts. And what we found out is that with like approximately 20 to 40 million reads, we were able to capture most of the KOs or genes that are like found in like gut microbiome samples. And then the annotation and QC part is really a tailored bioinformatic pipeline that's basically going to do a lot of QC step where you're going to remove the adapter, remove any host mRNA, then remove undepleted ribosomal RNA bioinformatically before doing the annotation through a 2-step process where you do a database pre-filtering and a database refinement that's going to be on a per sample basis for like best results. And I think as we were building those technologies and those kits, what we wanted to do is kind of think of designing a pilot that we could showcase the usefulness of gut metatranscriptomics, and also like showing like how different it is from like the typical go-to, which is gut metagenomics. And we looked at the existing literature and kind of saw a lot of papers that have been published on the impact of diet on the gut microbiome. Some studies have looked at like fiber intake or probiotics and how they can impact the community that's living within the gut of individual. Other studies have looked at vegetarian versus omnivore lifestyle and kind of compared like profile. And there were also a few studies that were very interesting to us that looked at the impact of intermittent fasting on the gut microbiome. And we were particularly interested by the intermittent fasting because this is something that's commonly practiced by many cultures throughout the world. And it's actually been associated with like major health benefits for the host. So like having a fasting component in your life is actually something that's pretty beneficial. And when we looked at the literature on like fasting and diet, we noticed that most of the work, and that was kind of expected, was based on metagenomics, so DNA-based sequencing. And they were able to show that diet can alter the gut bacteria and the community, but there were a lot of inconsistencies and different studies had different conclusion or different changes. So I think a lot of this can be explained by like suboptimal tools, study design, extraction, and so on. That's often a pitfall in the microbiome field. But one of the questions that we ask is DNA the most relevant analyte to look at kind of a dynamic community, and how changing the environment for those colonic bacteria by changing your diet is going to have an impact. So we recruited 7 healthy like subjects and we basically asked them to like follow a modified 5-2 intermittent fasting protocol. So the 5-2 intermittent fasting protocol is very simple. Every week, participants were asked to consume their normal diet for like 5 days, and then they were asked to like fast for 2 days out of the week. And we basically did that over a 2-week period. And we had slight modifications. So typically people don't do consecutive days of fasting, but in our case, we wanted to like make sure that we were going to capture the impact of diet on the gut microbiome. So we felt that consecutive days of fasting was better. And the other modification that we did was asking folks to fast for like 3 days that second week, because everybody tolerated that fasting for 2 days very well. And again, we wanted to like have the best chance of success. And throughout the studies, we collected a total of 70 samples from those 7 participants, so that's 11 by donor, that were kind of representative of either the normal or the fasting days. So they're highlighted in different colors so that we could basically compare the gut microbiome during fasting versus normal diet. And the insight I've got here is like just showing you the high participant adherence to the study that we had. So as you can see, this is a plot showing average calorie intake per day for all 7 participants. And you see that normal days, we're looking at approximately 1,800 calories per day on average. But during the 2 fasting period, you see like a significant drop. So we're talking about approximately 3-fold on average. And people were only consuming about 619 calories per day. So that's a significant drop. And just quickly, so we use OmniGene Gut and OmniMed Gut to like collect the samples. And to mimic real life conditions, what we had donors do is collect on the day of the collection in either of those devices, but then we held them at room temperature for 8 to 10 days to like kind of mimic the time it would take for like those samples to be returned to a processing facility. And then we did either DNA RNA co-extraction or like metabolite recovery for untargeted metabolite profiling. And as you can see, DNA RNA, very good quality. And we actually then put them through the Diversigen metagenomic and metatranscriptomic pipelines at 20 to 25 million reads per sample, and look at taxonomic and functional profile, as well as Kzyme and antimicrobial resistance gene. And for the metabolomics, we use the metabolome pipeline, so that's UPLC-MSMS, so mass spectrometry, to like generate those metabolomic profiles. So jumping on to the results of our like study. So the first thing that we looked at are like Kzyme annotation. So Kzyme stands for carbohydrate active enzyme, and they're essentially all the enzymes that are involved in the anabolism, modification, and catabolism of carbohydrate. This is a huge group of enzymes. We're talking about 5 classes and 300 families, and there's a lot of studies that have indicated that they're involved in every aspect of carbohydrate metabolism in the human gut. And as such, they're very, very important. I'm sure some of you have heard about short-chain fatty acid, or SFCAs. They're actually an byproduct of carbohydrate metabolism. And those SFCAs have also been shown to have like implication and to be involved in human health and disease state when it comes to the gut. So here, first slide, we compared like DNA and RNA profiles for like Kzyme annotation. So DNA is like basically the predicted Kzymes that are present in the genome. So keep in mind that we're not looking at expression data. And the one conclusion when you look at the DNA profile is that we're able to detect a number of different Kzyme in those metagenomes. But you see that relatively speaking, for all 7 donors, they're very stable over time. And we collected like 11 time points, and you really see that it's almost like a line, and there's not much change in the relative abundance of those Kzyme. What's drastically different when looking at RNA profile and metatranscriptomics, like you do see that there's a lot more variability. So generally speaking, there's not as much, the relative abundance changes like quite rapidly and is dynamic. And throughout the course of the study, you see some of those functions that increase or decrease. And I'm not going to dive too much into this, but highlighted in like pink, you can see the 2 fasting period that people went through. And there was not like a conserved change across the entire board. So we did see people responding to the diet and Kazan like changing, like going up and down, but it was not something that was consistent across the entire cohort. And we actually saw like some donors looking like they were responding to the diet and their Kazan were as well, but other donors didn't seem to respond despite those like changing and that dynamic profile. We also look at AMR, and I'm not going to be able to show that data, but for antimicrobial gene resistance, we saw like very similar results. So DNA profiles were highly stable, not much change happening over the course of the study, but with RNA, we did see log scale change differences day in, day out, but no true impact of the diet. So now diving into what happened in terms of like species level taxonomic profiles. So here we're looking at PICO plot and kind of the similarity of the samples. So again, we're comparing DNA versus RNA, so metagenomics versus metatranscriptomics. And when you look at DNA profile, you're not looking at expression data. You're looking at the relative abundance of the DNA over each of those species. And the results were exactly what we expected. You see each of the samples that were collected from the same donor tend to cluster together in those ellipses, meaning that the donors are like always more similar to themselves than they are to other donors. And generally speaking, if you look at the open symbol versus a closed one, we didn't see a strong correlation or an impact of the diet on those profile. As you can see, like there's kind of random clustering of the open and closed symbol for all of those donors, kind of meaning that diet didn't seem to like induce a change in the relative abundance of those species. When you look at the RNA, it's a very different picture. So first of all, you notice that the ellipses and kind of each time point, they're like bigger. So that kind of means that there's more variability and there's more dynamic changes. And that leads to like more dissimilarity for the samples that are collected from the same donor. You still see samples collected from the same individual kind of falling in the same region of the graph. But what's interesting too, and it's hard to like picture on this graph, is that you start to see some donors that show subclustering of the open and closed symbols, so like kind of fasting versus normal diet days away from each other, meaning that there seems to be consistent change that are like potentially driven by the diet. A better representation of this is kind of looking at the individual donor, because not every donor seems to respond the same way. And here I'm showing two examples. So donor 1112 is what we would call a non-responder in our study, where you don't see diet inducing consistent changes in the profile. So this is done through metatranscriptomic profile. And you do see that the points that were collected and each time point basically moves around that graph and is different from all the other one, but there's no pattern to it. And you kind of don't see like the fasting time point clustering together versus the normal diet days clustering with each other. Very different story for donor 1117, which is like a very strong responder, the strongest responder that we had, where you really see that normal diet days tend to cluster on the left side of that graph, whereas all the fasting days tend to cluster on the other side. And what's very striking is that we had that middle time point, so between the two fasting period. So if you like follow the graph, you see the first three from the top are like on the left side. Then for the two fasting days, it shifts to the right side. You're back to your normal diet. It shifts completely back to that left side again. And then second period of fasting, it shifts again to the left to then go back to the normal again. So there's very strong correlation between the diet and the profile as per like metatranscriptomic profiling. So obviously we were curious to like understand like what were like the differentially abundant KOs, so genes in those data set, both metagenomics and metatranscriptomics. And we looked at the one that were like differentially abundant in both DNA and RNA. And as you can see, very striking difference in the results. When you look at the DNA, there's very, very few functions that were found to be either higher or lower. But when you look at the RNA, you do see a lot more function. And the scale is the same for both graphs. So for like donor 117, you can see like upward of like 700 genes that were differentially expressed, either higher or lower. And what's also interesting is that with META-T, you see more of a gradient of response. With DNA, it looks like most folks are not responding to the change in diet. But with metatranscriptomic, you start capturing like more shades of gray, and there seems to be a gradient in the response of the participants. So donor 16 responds maybe a little bit, donor 15 a bit more, and then 14 and 17 show the strongest response, while 11 through 13 don't show any response at all. So we looked more into like those differentially abundant functions, and here it's just kind of the same data represented differently. So it's a heat map, and again we're comparing metagenomic, so DNA versus metatranscriptomic, the mRNA, and kind of looking at the top 100 most differentially abundant function in both the DNA and the RNA, and that's for donor 17. So I've kind of put boxes. Sorry about that. Lights are on a timer. That's unfortunate. But when you look at the heat map for like DNA, you do see some of the relative abundance of those functions that changes over time. However, those changes are a bit random. They can increase, decrease, and it seems to be highly dependent on the day of the study. So from top to bottom, it's T1 all the way to T18. And there's no pattern that seems to like correlate with the shift in diet. So the first 3 rows are normal diet, and then you've got 2 fasting days, and then you enter the week 2, like similar thing. There's no correlation. Very different story when you look at the same function with metatranscriptomics. So you do see that all the functions that are on the left side of that heat map are like very highly expressed during the normal diet days, and the other half of the function, so the right side, they're dark, so they're not expressed during normal diet. But as soon as you collect samples during the fasting, you see a complete reversal of that pattern. So all the normal diet days function get turned off, and a new set of function in response to like the fasting gets turned on. Then we've got that intermediate time point where people were back for a few days to their normal diet. The pattern reverses, and very nicely, as we go back to that second fasting period, it reverses again. So very, very strong correlation between diet and functional output for like metatranscriptomics, while the DNA profile, like you don't see any of that. So we ultimately asked a biological question, and what we wanted to do is try to like understand what those changes were and what they meant. And we started looking at, again, donor 117, just because of the scale of the response of that particular individual. And we looked both at overrepresented genes or like functions, as well as overrepresented function. And what was interesting is that, first of all, we didn't see any consistent changes seen across all the participants, but when we started looking within each of the participants, we noticed that there was an overrepresentation of amino acid and protein metabolic pathways that were either upregulated or downregulated. You'll see that for the most part, so those graphs are like kind of showing you like main pathways and the number of species that either down or upregulated. You can clearly see that by far, like the strongest response we were able to detect was like ribosomal function kind of being shut down for like upward of 60 species, and then a number of amino acid or like protein related pathway being shut down in like 10 to 20 species. What I think is also interesting and intriguing about this data is that we did see with metatranscriptomics like response that's at the species level, so biosynthesis of amino acid was upregulated for 6 species and downregulated, I believe, for approximately 20 or 23. And what's interesting is that it kind of shows you that different species respond differently to the change in the environment, so that fasting. And some species seems to thrive. They basically express more of those genes, while other people seems to like shut down. So we also looked at the metabolomic profile. So this is very early data, and unfortunately, I'm not going to dwell too much on it because we're still like working our way through the analysis. But we did see like very similar results as what we did for DNA and RNA-based profiles. So metabolomics profile are highly donor specific. So you can see here each sample is collected from each donor tend to cluster with the samples from the same donor. And generally speaking, we did not see a response that was consistent across the entire cohort. So again, we were in a scenario where each donor responded differently to the fasting challenge. One of the most consistent change that we did see was around like secondary bile acid metabolism. As you can see here on the like figure that's on the right side of my screen, so heat map, it's showing that secondary bile acid metabolism was downregulated for 5 out of 7 donors. And what we believe is happening here is that bile excretion is tied to the cholesterol intake. And during the fasting period, our like volunteers were like told not to consume any meat. So they were basically only consuming vegetables and fruits. So what we think happened here and why it actually makes sense is that we decrease the intake of like meat protein and like cholesterol, and as such that decrease like the excretion of bile acid in those individuals. We also did pick up like different donors kind of increasing like tyrosine, leucine, so kind of the same theme around amino acid and proteins. But as you can see, like each donor is very individual, and a lot of those changes were not super consistent. So trying to understand the biological response that we did see and what does fasting actually do to like the gut microbiome, I think what's interesting is that we tend to approach nutrition from the standpoint of the host, so ourselves. So like, you are what you eat, and if you eat well, you're going to be healthy. I think we tend to forget that the gut microbiome plays a crucial role, and that everything that we eat doesn't actually make it into the distal part of the colon. And the main nutrients that are actually available in the distal gut, so for colonic bacteria to use, are mainly undigested fiber and carbohydrates, so the carbohydrate that we don't have enzyme to process, and also a lot of unabsorbed amino acid and proteins. But everything else tends to be digested and uptaken by the host very early on during the digestion process. And we started to look at the average micronutrient intake across all donors and all days of the studies. And as you can see, generally speaking, like the 2 fasting period are like circled. We do see a drop in every micronutrient. So there's a drop in carb, protein, fat, not so much fiber intake. And that's essentially expected because you're restricting the calorie for those donors. But what's interesting is that the most drastic decrease was really around like protein and fat. And based on the fact that we know that fat is going to be absorbed by the host in the duodenum, we kind of hypothesize that maybe like the impact that we did see and all those changes around amino acid metabolism were basically driven by the fact that through fasting, we limited the bioavailability of amino acid in the distal gut. And we did a few search on PubMed, and we actually came up like a number of like seminal studies in like pretty high profile papers that have shown that nitrogen is actually the rate limiting nutrient in the gut of mammal, and that amino acids and proteins are like the primary source of nitrogen in the distal gut for all the colonic bacteria. And if you look across the board at different herbivores or different animals, whether they're herbivores or carnivores, generally speaking, the C/N ratio, so the ratio of like carbs to protein, is actually there's an excess of carbs and like a limiting amount of nitrogen. So presumably all these bacteria are kind of fighting for that limited supply of nitrogen that comes from protein. And there's tons of bacteria that have been in the past characterized and shown to like rely heavily on amino acid catabolism for energy production and go about their business. One such example of that is Clostridia, which is like a taxa that's very important for the gut. Where it gets complicated is that when you start like going down that rabbit hole is that different bacteria have different strategies to like store their nitrogen and amino acid, and some have actually adapted to use the host nitrogen. So we secrete mucin to like protect the lining of our like gut, and some bacteria are actually very good at degrading the mucin to get the amino acid that they need. Then the second source is obviously dietary nitrogen, which will be impacted by fasting. And the third one, where it's complicated, is that there's also a lot of cross-feeding. So every bacterial cell technically has all the genes that they need to produce any amino acid that they want. But it turns out that in reality, like bacteria tend to be strategic. And there's like keystone species that, for example, produce like a lot more protein than they need. And they excrete the excess so that other bacteria use that. And another bacteria is going to in turn produce a specific amino acid. But what we really believe is that by limiting the bioavailability of nitrogen and protein in the diet of the individual, we basically like force those bacteria to adapt, and we limited the amount of amino acid that they had. And essentially all the species that primarily rely on dietary nitrogen kind of were forced to like shut down during those fasting days. In summary and conclusions, I think what we've shown is that intermittent fasting like induces like personalized response, and the gut microbiome of different individuals don't respond the same way. What's intriguing is that you're able to capture like very similar taxonomic and functional features in DNA and RNA. So metagenomics and metatranscriptomic sequencing are similar. But when you actually look at the differences between the two, RNA is far more dynamic, and it also like captures functional changes that are completely missed by MGS, because MGS is only like giving you an idea of what could be expressed. It doesn't have like true expression data. Our metabolomic analysis so far like has turned out to be inconclusive. We're still working at it, but it seems that the best and the most conclusive data really came out of metatranscriptomics and RNA. I think the heterogeneity in the response that we saw to like the fasting regimen is presumably like rooted in the unique community that each donor has. So as kind of mentioned at the beginning, like every donor is very different from every other donor. And as such, they have like unique bacteria that may react differently. And then there's that community effect where different communities may be like more or less resilient to deprivation of amino acid through like diet. Other confounding factors, so this was a pilot study. I think we learned a lot as we were running through the process and also the analysis. One thing that we didn't really consider strongly in the early stages was the transit time. We kind of assumed that a sample collected on day 1 was representative of the day before, so day 0. It turns out that we've since gone back to those donors and measured the transit time, and this is actually not the case, and that could be a huge confounding factor. And interestingly enough, we actually noticed that the 2 donors that showed the greatest response were actually the people that had the fastest transit time. So we're talking about 36 to 40 hours on average, whereas a non-responder had the longest transit time. So that's very intriguing. And as I kind of mentioned, other things that are potential confounding factor are that bacterial cross-feeding and anything that may not be regulated through transcription. So lesson learned and future directions. I think what's great is that we've built and demonstrated that there are tools and technologies that are available to look at the gut microbiome through like a multi-omic lens, and not necessarily stick to DNA, because that's the way we've been doing it for a long time. We're continuing to explore this data in a multi-omic and precision medicine context. I think what's interesting is what is driving that individual response to fasting diet, and can we identify a marker that can help predict a response? I think as human beings, everybody care about their diet, and we've all tried like different diets to feel better, to be like, more active, but doesn't mean that the diet that you choose is necessarily well suited for you. And some of the data suggests that maybe some people are actually going to respond well to intermittent fasting, while other won't. I think one of the big outstanding question is also around which analyte is going to be best to like study the response of the microbial gut community to like a challenge or a modulation. And that's a tricky question because I think it ultimately depends on the question and the study. And we're really like trying to like leverage all those lessons and everything that we've learned through the process to like start thinking of future studies and like really having like an optimal study design and technologies. And I think the theme that we see emerging is that you want to normalize and standardize as much as possible. Like people are very different, they eat very different things, as such they have very different communities. And I think if you want a chance to like see commonalities in the response, you would want to normalize and standardize the diet as much as possible. I think we also want a better grasp of the baseline. So in our study, we only had a couple of baseline sample. I think given the huge variability that we've seen in metatranscriptomic dataset, we would have benefited from more samples to like kind of correct for that noise, and also the timing of the collection. So back to that comment around transit time and making sure that the sample that we collect are actually representative of what we think they are. And then the last thing is, I think, better understanding of the meaning of the changes through like different analyte. And as you can see on the right side of that slide, I think we need to start thinking of what each of those analytes really mean. So I think with DNA, you're really dealing with composition. So bacteria that are present and that have a membership in that community, but it doesn't give you any idea of who's active and what they're doing. RNA on the other side is going to give you like view into like the species that are active, and actually also the species that are like occupying like a given ecological niche. So they have the right set of genes to process like, you know, nutrients that are in the environment, and they're taking advantage of it and kind of like moving into that spot. While similar species with similar genes actually are dormant because they're waiting for their sun, their spot in the sun, or like the right environment. And then metabolomics, in general, I think you have to keep in mind that it doesn't give you any idea of the species that are active or any species level insight. It's basically the raw biological output of the entire community, and we're looking at byproduct of microbial metabolism. So changes in metabolomics are presumably driven by changes that are shared by all the species in the sample. Otherwise, it might be harder to like detect changes at the metabolomics level. So that's all I've got, and thank you very much for your attention. I'll be more than happy to take a few questions.
Okay, fantastic. Thank you for that presentation. It was really interesting. I think everybody learned a lot. I know I certainly did. So we've got a couple of questions already from the audience, and just a reminder, people can continue to throw those in the chat bar, and we try to get to them. The first question is one that I think you sort of touched on towards the end, but it's an important point, I think, so it's worth revisiting. So this individual asked, what could be the potential reason that RNA profile of some donors shows a response on their gut microbiome, but some have a greater effect on that of their microbiome? So I think really the idea of responders versus non-responders.
Yeah, it's a tricky question, and I think short answer is going to be we don't know for sure. We have hypothesis. I think part of the reason might be rooted in the community, in those donors. So presumably people may consume different amount of protein on a daily basis, and somebody that would consume a lot more protein than somebody else may have more dependency on those proteins, or their gut bacteria do. So part of the reason could be like rooted in like lifestyle, and basically your gut community, I think, is a representation of that lifestyle. But there's also a lot of potential confounding factor, as I was kind of mentioning, like transit time was unknown when we ran the pilot study, and we've noticed that the donors that didn't respond had a longer transit time. So did we actually collect samples that were not relevant and kind of representative of the normal diet days? It seems that there's a potential. And I think the last thing is maybe just around the bacteria themselves and their resiliency and how they interact with each other and how resilient they may or may not be to change in the environment and deprivation of amino acid or proteins. So we're still trying to like understand, and we're thinking of doing like a follow-up study to like answer all of these questions. But it's a fantastic question.
Yeah, it's very interesting. So we have another, I think, really excellent question here. So this person asks, can the mRNA expression profiles be used to classify patients in clinical trials that evaluate effects on the microbiome? And if so, how easy or feasible do you think it would be to do that?
Great question. Very tricky to answer again. Like we have limited data, like this is only like 7 donors. I do believe that mRNA expression profile could definitely be used in like clinical trials. If like the intent is to measure the impact that like the treatment is going to have on the gut microbiome. I think the problem to like really tease that out is that we would need like fairly large cohorts to kind of correct for that variability, and then try to like see if there's an emerging team for like, you know, people that respond or don't respond well to like a particular treatment. So I think the jury ultimately is still out on this. But I do think that what's promising about mRNA is that it's much more dynamic. And I think there's like a much faster change to like whatever's changing in the environment. So I think there's potentially like a better ability to like tease out like what a clinical trial and the drug that people are taking is actually doing to gut bacteria and DNA.
Yeah, definitely. So I have a question of my own, actually. And this one's a little bit more on the technical side. So I know when you want to do differential expression, usually you'll have to standardize the samples to make sure that you're comparing like versus like. But I'm not really familiar with how that works on the scale of a microbiome sample, as opposed to say like a cell culture where you're dealing with more of a defined population. You can just use a housekeeping gene or something like that. So I was just curious to hear maybe a little bit about how you manage to standardize across those samples.
Yeah, another great question. So I think one of the difficult thing about the microbiome field in general is that you're always dealing with relative abundance data. And as such, you can never normalize because you can never assume that there's something that's not changing, right? So the only way to like really do those comparisons is to like look at changes in that relative abundance. And there's actually no solution for absolute quantification of those changes or like how active or like prevalent certain bacteria may be with like DNA or RNA. So it's very tricky, and I think it's important to keep in mind that it's always relative because of the data that's generated by microbiome study and its nature.
Mm-hmm. Yeah, okay, that makes sense. It's one of those tricky things that I think we just have to accept when we're working with such a complex population. It's much more difficult to do certain basic things. I think we have time for one more quick question, unfortunately also coming from me. So I was really struck when you were showing the DNA for the caseins and how really consistent it was across the different individuals. So I was curious if you had any idea about why that might be, and if there were other kinds of enzyme families that would also be very consistent across different patient samples.
So it's not unexpected in the sense that a lot of people have looked at taxonomy on like longitudinal like studies and shown that when you look at DNA, generally speaking, like people microbiome don't change much over time, so they tend to be fairly consistent. Not that there's no changes, but it tends to be like fairly consistent. So that's expected. And I think when you look at the Kzyme, keep in mind that with DNA, you're not looking at expression, but the presence in that like genomic DNA from that particular species. So you would need like the species to have like 10 times the amount of cells to like really see a big shift, right, in the relative abundance as per DNA. Whereas RNA, you may have like one copy of a gene, but you can transcribe it 100, 1,000 times. So that's going to change much more rapidly than DNA. So I think when you really think about it, that our data makes plenty of sense. And I think it also really highlights that DNA-based sequencing has limitation in your ability to like really capture those like more subtle changes and those more rapid changes.
Right, okay. Thank you for that. So with that, I think there are no further questions and we're almost out of time. So I think we can start to wrap up now. So of course, I want to thank Bryce for his time, for coming in and giving us this really excellent presentation and answering the questions that we had. And I'd like to thank everybody for attending. So as a reminder, of course, this webinar was recorded and it will be shared with all attendees and registrants via email before the end of the week. If you're interested in finding out more about DNA Genotek or Diversigen, you can follow them on LinkedIn, or you can visit them at either dnagenotech.com or diversigen.com. Of course, on our end, you're more than welcome to follow DrugBank on LinkedIn or visit our website at drugbank.com. We'll have more of these webinars in the future, so we hope to see you at a future event. Thank you, everyone, and have a great rest of your day.
Thank you.